For Israeli and Goldenfeld, it's all about taking one of two possible paths and getting to the same place. One path says, okay, look, I have a fine-grained description of the system. I'm going to evolve it forward with my fine-grained rule. And at the end, I'm going to simplify the answer. I'm going to say, look, yes, I kept track of all these details, but in fact, you know what? I only need to know some of the output. I only need to know some of the final state of the system. So you can think of that as sort of walking along like this and then projecting up. Another way you can do it, though, right, is you can say, look, you gave me this fine-grained description, but I know I don't really care so much about that, so I'm going to project that down. Here's my fine-grained beginning initial condition. I'm going to project that down to a simpler description, and then I'm going to use a new rule that allows me to evolve that simplified description forward. So there are two paths, and if you've done it right, they'll get you to the same place. Evolve the fine-grained system forward and project, or project and evolve the coarse-grained description forward. A mathematician would say that these two operations, right, the operation of evolving forward and the operation of projecting up, commute. You can do one or the other in either order and you'll get the same answer. A then B, projecting then evolving, is the same as B then A, evolving then projecting. So let's see how this plays out with a particular example. And again, I'll just take one from their paper. This is Rule 105. Rule 105 is quite similar to Rule 150, which you've seen before. It takes the XOR of the 3 pixels above the pixel in question and then inverts it. So that's the only difference between Rule 105 and 150 is that final inversion. Another way to think of that is equivalently, the output is black when there's an even number of black cells above. All right. So now you know what we have to do, right? The first thing is we're going to consider not the final state of 1 pixel, but the final state of 2 pixels. And we're going to ask what happens not when you take 1 time step, but in fact when you take 2 time steps. And that means that those 2 final pixels will depend on a group of 6 pixels, 2 time steps previously. And now we'll consider those pairs of pixels to be these supercells. So you have a big supercell here, which is 2 pixels, takes on 4 possible states. And you have 3 supercells up here. So that's our F hat. What we have to do now is find a combination, a projection, P, that takes that supercell and summarizes it, simplifies it. It maps each of those 4 possible states down to one of 2 possible states. So we have a projection P, and we want to find an evolution operator G that allows us to evolve forward those projected down superstates. So the P, right, is what takes you from the fine-grained description up to the coarse-grained description, and that G is what's going to take you between 2 coarse-grained descriptions at different times. So you can either go F, 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 P, or P, G, G, right? For every 2 times you iterate F, you're going to, of course, only need to iterate G once. In this simple example, we'll just do the case where you skip 1 step, and so you have supercells of size 2. So, fortunately, it turns out it is possible to find a P and a G that enable that diagram to commute. And here it is in the case of Rule 105, right? In this case, the projection rule says, look, if the supercell has 1 cell that's black and 1 cell that's white, make that cell white. If both cells are white or both cells are black, then make it black. It's sort of like an XOR rule itself. In fact, actually, it looks a little bit like an edge detector. It says, look, if there's a difference within the supercell, mark it one way. But if there's no difference, if the supercell is sort of homogeneous, mark it the other way. If you use that projection operator, then it turns out, in fact, that your G, right, which is now, of course, taking binary values, right? Take a binary value because you projected the 4 possible state supercell down to a cell that only has 1 of 2 possible states, and that's what G operates on. That G evolution operator actually turns out to be Rule 150. So what we've shown is that it's possible to find a non-trivial coarse graining. It's actually quite an interesting one. It's possible to find a non-trivial coarse graining and an evolution operator that's still within the space of cellular automata. It's possible to find an evolution operator that enables that diagram to commute. And so now, just as we were able to talk about different kinds of Markov chains coarse graining into each other, we're now able to talk about how rules coarse grain into each other. And in fact, for a non-trivial projection operator, Rule 105 coarse grains into rule 150. Here's what it looks like. In the top, you can see the fine grain level description, and in the bottom, you can see the coarse grained level description. At the top there, you can see that we have these smaller pixels, and those smaller pixels are both small in the x direction along this axis here, and smaller in the time direction than in the coarse grain case. The coarse grain case, of course, lumps pairs of states into one, and then in fact the jumps are now larger. They're 2 time steps instead of 1. And by looking at the comparison between these two, you can sort of see what's going on, right? First of all, of course, the coarse grain description is capturing something interesting about the fine grain description, right? We still have this idea that these triangles, these sort of these little perturbations that we begin with, that lead to these expanding waves, right? We still get that kind of wave-like picture, right? These sort of propagating spaces that have kind of internal structure. But you can also see that we're missing things too, right? So if you look at those 2 triangles at the fine grain level, one of them is sort of darker than the other. But in fact, when we coarse grain, the differences between those 2 triangles goes away. So somehow, rule 150, when we evolve it forward, is operating on a coarse grain description that's thrown out some interesting features of rule 105. Another obvious feature here that distinguishes rule 105 from rule 150 is, in fact, we lose that kind of zebra stripe pattern, right? And that's, of course, because if a pair of squares is both white or both black, the projection operator maps them both into a square that's both black. So we've lost some of the structure, both in the sort of places where those propagating triangle waves haven't reached yet, and also within the triangles themselves. So this gives you a little bit of a better sense. Now, as we'll see, it's not always the case that the picture that you get when you coarse grain looks similar in some important respects to the fine grain description. Here, it's a particularly elegant example of how we were able to capture something about the rule, but of course, not everything. We can't really capture everything, of course, because that projection operator is a lossy compression. It throws out information. And for rule 105, it really matters whether everything is white or everything is black. But in fact, for rule 150, when we do the projection, it maps both of those cases to the same state. So, Israeli and Goldenfeld hacked, and they hacked and hacked and hacked, and they looked at all 256 rules, and they tried to figure out how, or the extent to which one rule could coarse grain into another. So these arrows here show you how it's possible to find, or whether or not it's possible to find a projection operator and an evolution operator that allows one rule set to map to another upon that coarse graining. And in fact, they consider not only supercells of size 2, but also size 3 and size 4. And computationally, this starts getting really hard because there's so many different kinds of projection operators you can use, and there's so many different possible evolutions that you can pick, that you start to run out of time. It gets exponentially hard to find a good projection operator. It gets exponentially hard to search this space. There's only a partial map. But what you can see here is, for example, in the bottom, the result that we just talked through a little bit laboriously, which is the fact that it's possible to find a projection operator that takes you from state 105, or from evolution rule 105, to evolution rule 150. By the way, one of the things you can notice from this graph is that it's clear that Israeli and Goldenfeld haven't actually found every possible coarse graining relationship. And that's because there should be a feature of this network that doesn't actually happen. And that's that if A coarse grains into B, if A renormalizes into B, if it's possible to find a projection and evolution operator that take A into B, and B renormalizes into C, it's possible to find a projection that takes B into C, then it should also be possible to renormalize A into C. Of course, now you're going to be coarse graining twice, and it's harder, of course, for Israeli and Goldenfeld to find those. But if you look at this chart, what you should see is, for example, the fact that not only does Rule 23 coarse grain to Rule 128, and not only does Rule 128 coarse grain to Rule 0, but it also should be the case that it's possible for Rule 23 to coarse grain all the way down to Rule 0 just by doing 2 projections and zooming out even further. That said, Israeli and Goldenfeld did a pretty good job looking at an enormous number of possible relationships between all of these possible rule sets. And I find these diagrams quite compelling. It tells you something really complicated, really interesting about how deterministic rules and deterministic projections map into each other. One of the things that you'll see from that network is that not only does Rule 105 coarse-grain to Rule 150, but in fact, Rule 50 coarse-grains into itself. So the pretentious way to say that this is a fixed point of the renormalization group, right? With that projection operator, you actually take Rule 150 into a zoomed out version of itself. Right? You sort of skip a step, you project down the supercells, and you recover the same rules. Now, it's important to notice there's a subtlety here, right? It doesn't mean that the image itself is self-similar. It doesn't necessarily mean that Rule 150 is kind of fractal in some interesting way, right? Because the coarse graining may not be the kind of coarse graining that just simply zooms out. Consider, for example, the projection we had going from Rule 105 to 150. That wasn't a simple decimation in the way that we did on the Alice picture, for example, at the beginning of this renormalization module. In that case, right, when we were renormalizing Alice, we took her picture and we looked at little packages of cells and we just picked one of the values to define the value of that grid, of that larger grid cell, that supercell in the Alice case. But if you remember the Rule 105 to Rule 150 projection that worked, in that case it was actually an edge detector. If the cell was all white or all black, it got mapped to something that was all black. So it doesn't necessarily mean that if you kind of fuzz Rule 150, it still looks like Rule 150. It really depends upon the details of that projection operator. That said, in fact, you might think of it another way, that Rule 150 is a fixed point of the renormalization group with potentially a much more interesting projection than a simple decimation.